If you are into medical imaging, deep learning, and you are up for a challenge, well, then we just got the right thing for you here today. Introducing the MIDOG challenge. Okay, first things first. In case you wondered, no, MIDOG has nothing to do with a my dog or my cat or any other animal. It is just an abbreviation for Mitosis Domain Generalization Challenge. That's it. In case you are not familiar with microscopy images, here is what you need to know. To assess the malignancy of a tumor, the current method of choice is taking a thin slice of tumor tissue and putting it under the microscope. There, the pathologist will see lots of cells. Some of them are normal cells, while others are cells that are in a state of cell division or mitosis. The occurrence rate of mitotic figures is indicative of tumor growth and thus of great interest. Now, if mitotic figures are that important, then of course the question is, why don't we apply some deep learning to it so that we can facilitate the workflow of the pathologist and with that maybe even have an increase in accuracy, which would of course benefit the patient. Now, of course, this idea is not new and people have tried this and actually have shown quite some nice results. So yes, in fact, this is possible, and it is possible even with an accuracy of the deep learning model that sometimes exceeds the trained pathologist. Now then, of course, the question is, why don't we use that in clinical practice? And one of the answers to that is that it might be related to images from different pathology labs looking different. Now, many people have attributed this to a difference in staining protocol. But one thing that has not been investigated so far is the change of a digital image when acquired using different microscopy scanners. So the same slide, when digitized with one scanner, can look totally different compared to when it was digitized with another scanner. You don't believe me, do you? Well, I can show you some examples. So first of all, here we do have an example of an image digitized with the Hamamatsu XR scanner, and down here we have the same image but digitized with the Aperio ScanScope CS2. And I think now you agree with me that this is quite some difference that we see here in these images. The training dataset consists of 4 scanners. Here we can see random crops from all scanners. And even though we calibrated for the ICC profiles wherever these were available and converted it to standard RGB colors, the differences are quite striking. So we got 4 scanners in the training set, each with 50 images representing 50 cases of breast cancer. All cases were taken from the diagnostic archive of the University Medical Center in Utrecht, the Netherlands. Out of those 4 scanners, only 3 have annotations for mitotic figures, while 1 dataset can be used for data-driven domain adaptation and generalization. This last scanner, scanner D, will also be part of the test set, and so is scanner A. Scanners B and C, however, are not part of the test set, but they are images acquired by 2 more scanners that we don't disclose at this point in time. As the name indicates, the goal of the challenge is domain generalization, that is, to work on methods that work independent of the scanner. I hope that I caught your interest now. If that's the case, then please become a participant and be part of the challenge. Thank you for watching this video, and thanks to all my collaborators on this challenge.